Hello everyone and welcome. In this video we're checking out question paper 12 February March 2025. Before starting, make sure to check out the free notes I have available for you, and let's go ahead and get started. Question number 1. In an electron micrograph, the length of a mitochondrion is measured as 117.1 centimeters. The magnified electron micrograph is 38,000. What is the actual length of the mitochondrion? So to measure the actual length, you just need a triangle, which is pretty much the question. So basically the triangle is going to be M, I, and A. Okay, so magnification, image length, and actual length. Now, magnification here is given it to you as 38,000, and that should equal to the image length, which is given to you as 17.1 centimeters. But because all the answers are in micrometers, I'm going to convert centimeters to micrometers. To do that, first convert centimeters to millimeters, which is by multiplying by 10, and then convert millimeters to micrometers by multiplying by 1,000. So you should get 171,000. Divide that by the image length, which we want, the actual length, which we want to find. And so here you're going to get the answer as 4.5 micrometers. There is a theory that mitochondria and chloroplasts were originally free-living prokaryotes. It is thought that millions of years ago, these free-living prokaryotes were taken into larger cells by endocytosis, the endosymbiotic theory, where instead of being digested, they become functional organelles. Which features of mitochondria and chloroplasts support this theory? Okay, again, this is a theory, it's not a fact, but because mitochondria and chloroplasts have 70S ribosomes, because mitochondria and chloroplasts have circular DNA, this was the first thought, and then these things make it there's a theory. It's not really proven yet. Let's see the options. 1. Mitochondria and chloroplasts are surrounded by double membranes. Okay, yes. Mitochondria and chloroplasts are surrounded by double membranes. Now you might think bacteria or prokaryotes do not have anything membranes, right? So they do not have any membranes, they don't have any kind of membrane-bound organelles. So why would mitochondria and chloroplasts having double membranes make them similar to prokaryotes? Because since these bacteria or prokaryotes were taken into the cell by endocytosis, so this is the cell, and then this is the bacterial cell, which has a single membrane, and then some DNA inside. Okay, what happens is that this is engulfed by the cell, meaning it's wrapped by a membrane, right? So the membrane sort of surrounds it, and then it basically comes in as a little vesicle. So this is endocytosis. So what we expect is that there will be an outer membrane, which is the endocytic membrane, and then there will be the inner membrane, which is the membrane of the prokaryote. So it makes sense that the organelles would have double membranes. Mitochondria and chloroplasts have small circular DNA. As we've mentioned before, mitochondria and chloroplasts synthesize proteins 1, 2, and 3 of each cellular structure. Lysosome, mitochondria, and smooth endoplasmic reticulum. For lysosome, digestion of unwanted substances, that is true. Basically lysosomes are going to be having containing enzymes, or what we call hydrolases, which are going to be multiple different kinds. We're gonna have lipase and protease and all these different enzymes that can break down multiple different things. Okay, and they're specifically mainly found in neutrophils and in phagocytes because they have a great function in phagocytosis. Spherical sacs containing hydrolytic enzymes, so both of these things are true. Mitochondria, abundant in sites of active transport, true because mitochondria are responsible for oxidative phosphorylation and basically aerobic respiration. So they produce lots of ATP. So we need ATP for active transport, and that is basically why mitochondria could be abundant in sites of active transport, as well as ATP synthesis. What about smooth endoplasmic reticulum? Processing proteins, lipid production. Okay, smooth endoplasmic reticulum do not process proteins. It's a function of the Golgi apparatus, the rough endoplasmic reticulum, but definitely not the smooth endoplasmic reticulum. The smooth endoplasmic reticulum produces lipids. It produces your steroid hormones, which are, again, essentially lipids. Produces cholesterol, lipids. Produces, it also is going to have functions in detoxification, so it's going to be lipid production. And as for which option is more correct, the digestion of unwanted substances versus spherical sacs containing hydrolytic enzymes, since they're asking for function, this is more of structure. Okay, so you're going to go with B. Which cell components are present in typical prokaryotic cells? Cell wall, ATS ribosomes. They don't have ATS ribosomes, it's only available in eukaryotic cytoplasm. They do have a peptidoglycan cell wall. X-ray analysis of fossilized cells found in rocks in central India dating back to 1.6 billion years ago has revealed several features. One, the cells are up to 145 micrometers long. Okay. The cells are joined end-to-end to form filaments. The cells contain some internal structures. The cells are surrounded by a cell wall. Which two features taken together provide more support that the cells are plant cells? The most important features here are the fact that the cells are going to be surrounded by a cell wall. Okay. And that the cells are joined end-to-end to form filaments. Because 145 is a bit too long to be a plant cell. I mean, for example, this end-to-end to form filaments is really common in xylem, so I'll have to guess it's 2 and 4, which is D. The electron micrograph shows an organelle found in some cells of many multicellular organisms. Which row shows the structures that are expected to be present in the cells containing this organelle? So this is chloroplast, as you can see, with the thylakoid membranes and the starch granules and everything. So basically, you can expect this to be found in plant cells and some protoctists as well. Cell wall, centrioles, plasmodesmata. Okay, what do you find in plant cells? You do have a cell wall, that's for sure. You do have plasmodesmata, but you do not have centrioles, so it is D. What could take place during a hydrolysis reaction? Glycosidic bond is broken, a molecule of water is produced, a sucrose molecule is split into a fructose and glucose. So this is an example of hydrolysis. Hydrolysis is just the breakdown of something with the use of water. Okay, so you're using water to lyse things, okay? So a molecule of water is produced? No, okay? This is what happens in condensation. In condensation, you can imagine it sort of like, condensation is basically this like rain droplets that form, for example, on your windows. It's just water that forms on surfaces, right? So condensation also produces water the same way water would form on your windows if it's cold. And so, no, molecules of water are used in hydrolysis but produced in condensation. A glycosidic bond is broken. These two are examples, so yes, 1 and 3 only. C. Sugars with a ring structure can also have a linear structure. Which sugar molecules could be represented by the linear structure shown in the diagram? Okay, what do we have here? Well, here we have a 1, 2, 3, 4, 5, 6 carbon atom, which definitely could be glucose. And if you look at the structure here, it is very specifically D-glucose. Okay, so it's glucose, deoxyribose, ribose. Okay, deoxyribose and ribose are 5 carbon-containing sugars. And so they're pentoses, so they can't be this one over here. You can count the carbons and you can see there are 6. So it's really glucose only. Which statement about triglycerides is correct? Each triglyceride molecule is formed by combining 3 fatty acids with a glycogen glycerol. Okay, a triglyceride molecule contains 4 ester bonds. No, between each fatty acid and the glycerol, there's going to be a single ester bond. Okay, so because there's 3 fatty acids, okay, here, let's imagine this is glycerol. Okay, you're going to have 3 fatty acids. At each spot where we're going to have a bond between them, it's going to be an ester bond, and that's 3 bonds. Triglyceride molecules form a bilayer in the cell surface membrane of cells due to hydrophobic and hydrophilic interactions. It's not triglycerides, it's phospholipids. The ratio of oxygen atoms to carbon atoms is lower for triglyceride molecules than for carbohydrates. That is true. For triglycerides, it's rich in those, as you can see, carbon chains, and definitely much less oxygen. The only oxygen available is the one in the ester bond, and I think, yeah, it's basically the 2 atoms available in the ester bond, which is why it's D. Hydroxyproline is synthesized by the addition of an OH group to the R group of the amino acid proline. Hydroxyproline is a major component of collagen and has an important role in increasing the stability of its structure. Which statement explains why the addition of a OH group to the proline could increase the stability of collagen? Okay. Why the addition of an OH group increases the stability of collagen? Now, there could be multiple reasons. Within collagen is a triple helix, right, where every third amino acid is glycine, and there will be bonds between the AH chain of the collagen triple helix, and these bonds will be covalent bonds. Now, it could be that this OH group is part of formation of a covalent bond between hydroxyproline and any other amino acid. Let's see what we have here. So it strengthens the hydrogen bonding between R groups of the adjacent polypeptide chains, resulting in tertiary structure that is more resistant to heat denaturation. Between adjacent polypeptide chains, you don't have hydrogen bonds, you have covalent bonds. And hydrogen bonds will not really do anything to prevent heat denaturation, so we can cancel out A. B. It increases the number of sites available for the formation of hydrogen bonds within the secondary structure of collagen, resulting in more stable alpha helices. Absolutely not. When you see alpha helix and collagen, you cross out the answer because collagen does not have alpha helices, it has triple helices. Triple helices means it has 3 polypeptide chains wrapped around each other in a helix. Okay, this is quaternary structure, not secondary structure, and collagen does not have an alpha helix secondary structure. So B is cancelled out. C. It increases the formation of hydrogen bonds between R groups and water molecules, which will help to hold the chains of the collagen triple helix together by strengthening hydrophilic interactions. Okay, strengthening hydrophilic interactions is sort of new formation between R groups and water molecules. Okay, let's see D and come back to C. It strengthens the quaternary structure of collagen by providing more sites for hydrogen bonding between the R groups of distantly separated amino acids within the same polypeptide chain. More sites for hydrogen bonding between the R groups of distantly separated amino acids within the same polypeptide chain. Okay, within the same polypeptide chain, you're not going to get hydrogen bonds between the collagen, between the amino acids, okay? So you would not get hydrogen bonds between anything within collagen, period. So you can cancel out D for the same reason we cancelled out A. That would leave us with C. Now, the formation of hydrogen bonds between R groups and water molecules, so between the R groups and water molecules, I guess that could be it. I mean, does strengthen hydrophilic interactions. Part of collagen needs to be facing water. Yes, collagen is fibrous and it's not soluble in water, but the part that is facing water needs to be able to interact with it so that it doesn't coil up into itself and becomes non-functional. So I could pick C in this scenario. Moving. Plant cell walls are strengthened by cellulose molecules that are arranged in several layers. Within each layer, the cellulose molecules are arranged in the same direction. So yes, within each layer they are parallel to one another. Which row shows the bonds that hold adjacent molecules of cellulose together within each layer and the arrangement of cellulose molecules in different layers? Okay, so within the same layer, bonds that hold adjacent cellulose molecules together are going to be hydrogen bonds, and arrangement of cellulose molecules in different layers are going to be in different directions. Remember, between cellulose molecule, within the cellulose molecule, you're going to have glucose molecules, and these glucose molecules are going to be bonded through glycosidic bonds, which are covalent COC interactions. Okay, this is a covalent bond, but here between different cellulose molecules, so complete chains of cellulose, they're going to have hydrogen bonds between each other, and the arrangement of cellulose molecules in different layers are going to be in different directions and different orientations, and that increases the strength of cellulose. Okay, and that's C. Galactogen is a storage polysaccharide in some animal species. It is a branched polymer that is branched from beta-galactose monomers. Okay, which comparison of galactogen with another polysaccharide correctly summarizes one similarity and one difference. Okay, so glycogen and galactogen are both branched, but glycogen is not a storage polysaccharide in animals. Okay, let's repeat this. So galactogen is a storage polysaccharide, like, glycogen. It is a branched polymer that is formed from beta-galactose. So the difference between glycogen and galactogen is that it's formed from beta-galactose instead of alpha-glucose. Glycogen and galactogen are both storage polymers in animals, but glycogen is unbranched. Glycogen is very much branched. Cellulose and galactogen are both branched. Cellulose is not branched, okay? Amylopectin and galactogen are storage polysaccharides, but amylopectin is formed from alpha-glucose monomers. It is exactly true, okay? Here you can see which levels of protein structure are always involved in the active site of an enzyme. So always involved in the active site are going to be... Okay, so yes, definitely all of them, okay? Quaternary, definitely not, because some enzymes do not have a quaternary structure. Some enzymes are made of a single polypeptide chain. But primary, secondary, tertiary, for sure, because they're all going to affect the way the enzyme looks and therefore affect the specificity of the active site. And so it is B. Catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. Catalase was added to a solution of hydrogen peroxide, and the oxygen was collected in a gas syringe. The total volume of oxygen produced from the start of the reaction was recorded every 10 seconds for 1 minute. The table shows the results. Okay, so time here, 0, 10, 20, 30, 40, 50, and everything. And then total volume of oxygen produced, and you can see how it increases. And then here the difference was like 10. Here the difference was like sort of like more than 10, definitely like more of like 18. And so the rate is going to slowly start to decrease. Like, we're still producing oxygen, but it's at a much lower rate. Here you can see how the difference is only 1 from here to there. So what could be concluded from the results? The reaction stopped after 60 seconds and no more oxygen was produced. No, you can't conclude that. Maybe the reaction went on, but again, at a really low rate. Okay, the highest rate of oxygen production occurred at 10 seconds after the start of the reaction. The highest rate occurred 10 seconds after the start of the reaction. Well, we're not really sure about that, okay? Like, the highest rate could be anywhere between, occurred 10 seconds after the start of the reaction. Okay, let's get back to this one. It took more than 20 seconds from the start of the reaction for half of the substrates to be converted into water and oxygen. It took more than 20 seconds for half the substrates to be converted to water and oxygen. Okay, if you look at the 20 seconds, you have 40 being produced. And do we know that we've reached half? Okay, let's draw the graph of the rate against substrate concentration, or let's do time. Okay, so this is total oxygen. Okay, and then this is the time. So the graph of the material, like, plotted out in front of us, okay? So at first it was 0, and then it was 22 at like 10, and then it again became like 40 at like 20, and then it slowly started to increase, but not at this rate, and then we've reached sort of like a plateau. Okay, we're like 57, 58, that sort of thing. So the rate of the reaction is basically, this is total oxygen over time. So the rate of reaction reaches 0 at a certain point, okay, where like there is basically not much oxygen being produced, but that's after a while and after like the 60 seconds, okay? But the mean rate of the reaction between 20 and 30 was twice the mean rate between 30 and 40. Between 30 and 40, the rate of reaction would be 5 divided by 10, which is 0.5. Between between 20 and 30, it's going to be 10 divided by 10, which is 1. So that's exactly true. The mean rate of reaction between 20 and 30 was twice. So I think this is the answer because it's the most obvious. But let's actually review the others because they might be confusing. The highest rate of oxygen production occurred after 10 seconds after the start of the reaction. 10 seconds after the start of the reaction, we reached 22. But between 22 and 40, the rate is definitely increased. And the highest oxygen production occurred 10 seconds after the start of the reaction. Okay, we can't really be sure about that if we don't really have all the details. It took more than 20 seconds from the start of the reaction for half the substrate to be converted to water and oxygen. We don't really know half the substrate to be converted from water to oxygen. I mean, if you look at the 20 seconds, we still have like lots of oxygen being produced even after. Like there's 10, and then there's 5, and lots of product. So I can't really tell you that more than half the substrate. It's really not that obvious from the results. Succinic dehydrogenase is an enzyme that catalyzes the conversion of succinate to fumarate in aerobic respiration. Malonate is a reversible inhibitor of succinic dehydrogenase, and malonate reduces the enzyme's activity by binding to its active site. Malonate and succinate cannot bind to the active site at the same time. Okay, so it is a competitive inhibitor. He said here it's a reversible inhibitor and that it binds to its active site, which statements describe the effect of malonate on the activity of succinic dehydrogenase. So it's very clearly a competitive inhibitor, which means it's going to increase the Km value, but it will not change the Vmax. In the presence of malonate, Vmax can still be reached if the concentration of succinate is increased. That is true. Okay, malonate has no effect on the Km. It's not true. The presence of malonate, Vmax can still be reached with the concentration of fumarate. Fumarate is the product. Has nothing to do with fumarate. Malonate decreases the Km. No, it increases it. Okay, so let's explain this for a little bit. The effect of a competitive inhibitor on the Km and the Vmax. So if you plot a graph of the rate of reaction against the substrate concentration, as you increase the substrate concentration, the rate of reaction should increase because there's more enzyme-substrate collisions, and so the reaction should increase. Okay, up until you reach a certain point where the enzyme is fully saturated with substrate, and so it can't really do anything more because the active site is completely occupied. What happens is that if we introduce a competitive inhibitor, the competitive inhibitor is kind of attractive to the enzyme as well as the substrate. So the enzyme decides, okay, you know what, let's have some of the competitive inhibitor bind the active site as well. And so that it increases the amount of substrate you need in order to reach that maximum rate of reaction because the enzyme is distracted. So you need to make sure that there's more substrate so that it can actually overcome the effect of the of the competitive inhibitor. And that's basically why in the end we're going to reach the maximum rate of reaction, but it's going to be at a higher substrate concentration. So we reached it like right here for the effect of a competitive inhibitor, the substrate concentration has to be this much, okay, much higher. The Km value is the Michaelis-Menten constant. It indicates to you the affinity of the enzyme to the substrate. It is supposed to be the concentration at which, or substrate concentration at which you reach half the maximum rate of reaction. So this is the maximum rate of reaction, we call it the Vmax. Half the maximum rate is approximately right here. The substrate concentration where you reach that, so let's say this over here, this is the Km value, okay? The more substrate, or the higher the Km value that you need to reach just the half the Vmax, that means the enzyme has a lower affinity to the substrate, and that's exactly what we see right here. You need a higher Km to reach half the Vmax, which means the affinity has been decreased, which makes sense. Affinity is attraction. Attraction has been decreased because there's a competitive inhibitor in the mix. So that's why the answer is A. The diagram shows the dimensions of 2 blocks of agar. The diagram has been drawn to scale, and the blocks of agar were stained pink with the pH indicator. In acidic conditions, the pink pH indicator becomes colorless, and the 2 blocks of agar were placed in a beaker of acid at the same time. As the acid diffused into the blocks became colorless. What's the surface area to volume ratio of the block that completely becomes colorless first? Okay. This is sort of like just a calculation question. Remember guys, the higher the surface area to volume ratio, the faster the diffusion. So we're going to calculate both surface area to volume ratios, and then we're going to get the one with the bigger surface area to volume ratio. That's going to be our answer, okay? So the surface area of a cuboid in this case is going to be 2 times length times width plus 2 times length times height plus 2 times width times height. So everything times each other, okay? And the volume is going to be length by width by height, okay? So 2 times length by width plus 2 times 20 by 4 plus 2 times 4 by 30. And that's going to be 1,600 for this one. And the volume is going to be 30 by 20 by 4, which is 2,400, okay? Second one, it's going to be 2 times 10 times 5 plus 2 times 10 times 15 plus 2 times 8 times 15. And that's going to be 640 for surface area. And for volume, it's going to be 10 by 15 by 8, which is 1,200. Okay, 640 divided by 1,200 is 0.53 to 1. And then 1,600 divided by 2,400, that's going to be 0.67 to 1. And so the bigger one is this one, which is why we pick B. The graph shows how the rate of facilitated diffusion of substance X across a cell surface membrane changed as the concentration of substrate X increased. Okay. All conditions except for the concentration of substrate X were kept constant. The temperature was maintained at 15 Celsius. Okay, so the concentration of substance X was and how it increases the rate of facilitated diffusion. And here you could see in the beginning it was impacting the rate of facilitated diffusion because substrates could just flow freely through the channel, and then at a certain point saturation happened. And remember, whenever we reach at this point where there's a plateau, that means that the concentration of substance X is no longer a limiting factor, okay? It's no longer a limiting factor for the rate of facilitated diffusion. We have other factors limiting, and most commonly that factor is going to be the channels or the carrier proteins or whatever protein you're dealing with, okay? So for example, for enzymes, it's the number of enzymes, okay? So here we're dealing with facilitated diffusion, which means the number of channels, the channels are completely occupied, and so you're going to reach saturation. The rate of facilitated diffusion of substance X at Q will increase if the temperature is increased. Okay, so these are other examples. So the rate of facilitated diffusion of substance X at Q will increase if temperature is increased to 20. So the temperature is maintained at 15. Okay, and that was fine. I think 20 is okay for like a cell surface membrane because the temperature, the optimal temperature for our cells is actually 37, and so 20 is fine. It definitely should increase the rate of diffusion, okay, because substances will move faster. The rate of facilitated diffusion of substance X at P will increase the concentration of it will increase if the concentration of ATP is increased. This facilitated diffusion is a passive process, does not require ATP, so that's wrong. Okay, facilitated diffusion of substance X is increased as, okay, so the rate of facilitated diffusion of substance X at Q will increase if the concentration of substance X is increased. We can clearly tell, again, as I've mentioned, the concentration does not impact the rate of diffusion anymore. Okay, and what about option C? The rate of facilitated diffusion of substance X at P will increase if the length of time over which the rate is measured is increased. Okay, the length of time has nothing to do with this. In this case, the rate is always going to be calculated using the same formula, so it doesn't really depend on the length of time. It depends more on temperature, on the number of channel proteins. Yeah, that's pretty much it. Okay, so here you can see which molecules form a bilayer in the cell surface membrane. That's phospholipid. A nucleus of a body in a body cell of a species of fruit fly has 8 chromosomes. How many strands of DNA are present in the nucleus at the end of interphase? Okay, so first let's draw the cell cycle. So at the end of interphase means basically at G2, right? Because what is interphase? It's so let's start with mitosis. It's like really short right here, and then there's cytokinesis, right? But then we hop back into G1, and then there's the S phase, and then there's G2. Okay, so for G1, you're fine with 8 chromosomes just as well. During the synthesis phase, DNA is going to be replicated, so you're going to have twice as much chromosomes. So it's going to be 6. Actually, no, you're going to have the same number of chromosomes. The chromatin are going to be the ones duplicated, so it's supposed to be 8. So yes, by the end, how many strands of DNA? So the chromosomes stay the same, it's 8, okay? But let's not forget, the chromatin is doubled. So previously you had a single chromatin. Now you're going to have 2. And each chromatin is composed of 2 molecules of DNA, right? So 2 molecules of DNA, a double helix. So since now the number of chromatin is doubled, the number of DNA molecules is twice that. Okay, so if you have 16 chromatin, then you have 32 molecules of DNA. Okay, so focus on what they're asking you. And then next you have the photomicrograph shows a plant cell in a different stage of a mitotic cycle. 4 of the cells are labeled with a number to identify them. Okay, so you have 1, 2, 3, 4, and you could see the stages they're in. One of the numbered cells is within a main stage of mitosis in which the spindle begins to form. A second numbered cell is within the main stage of mitosis in which the spindle fibers shorten, which identifies the 2 cells in these 2 different stages. Okay, so you can see 1 is metaphase, 2 is anaphase, 3 I think is like prophase, the beginning of prophase, and then 4 is like by the end of prophase, because here you don't really have a nucleus, so I can't really say it's like before mitosis, because they also very specifically said they are within. No, it could be definitely a cell with the nucleus intact. Okay, we can't really judge. It's a light microscope anyway. Okay, here you could see the chromatin are very clearly condensed, so this is prophase. Now when spindles begin forming, that's going to be in prophase, so you could say 4, and then when anaphase is happening, that's going to be 2, so that's D. Which percentage of the chromosomal DNA present in a cell during G1 is present in the same cell later in the same mitotic cell cycle during prophase and during telophase? Okay, so the percentage of chromosomal DNA in a cell during G1, okay, so during G1 is present in the cell and the same cell later in the same mitotic cycle during prophase and telophase. Okay, so during prophase and telophase, during prophase, you would still have the cell, like, having the same percentage of the chromosomal DNA, right? It will still have the same chromosome, it's just going to be duplicated. And same thing for telophase. The cell still hasn't split yet, and even after it splits, the chromosomal DNA in cell 1 is going to be the same as that in cell 2. So I have to say it's 100%. The statements are about 2 genes and their protein products that have a role in tumor formation. The protein coded for by the PTEN gene prevents cells from growing and dividing too rapidly. The cell coded for by the P53 gene prevents cells progressing through mitotic cell cycle if they have damaged DNA. Which combination of mutations of these 2 genes in an individual is most likely to result in the formation of a tumor? Okay, this definitely results in the formation of a tumor if it's mutated, because the P53 gene should stop the cell from undergoing mitosis if it's damaged DNA. But if the cells do mitosis either way, then they could definitely start dividing uncontrollably and everything. Whereas the protein coded for by the PTEN gene, which prevents the cells from growing and dividing too rapidly. So this gene is important. Now, if it's mutated, then what happens is that the cells are going to start again dividing really rapidly. So 1 and 2, mutation present results in tumor formation. Telomerase is an enzyme that prevents the shortening of telomeres. It's not present in most normal cells, but is active to an estimated 85% to 95% of human tumor cells. Which statement explains the effect of telomerase on human tumor cells? Telomerase triggers a self-destruct process known as apoptosis, ending the life cycle. Not true. Telomerase helps the cell live longer by increasing the length of telomeres. Telomerase damages the chromosomes so they become genetically unstable and are unable to replicate and divide. Wrong. As I've mentioned, the increase in length of telomeres, so as the cell can pretty much divide as many times as it wants, the DNA can replicate as many times as it wants without having to worry about the fact that DNA polymerase, when it comes to replicate DNA, is not going to run until the end of the chain of DNA, which means some of the DNA is going to be lost. Now, because this DNA, which is telomeres, does not code for anything, then you're fine. Until the moment that you lose the telomere, the cells cannot divide anymore. The cells are not allowed to divide anymore. Telomeres help humans, tumor cells, avoid senescence or cell death, which is usually the expected consequence of repeated cell division. Exactly. It helps to avoid cell death, which is also known as senescence, which is usually the expected consequence of repeated cellular division. Which diagram correctly represents one of the base pairs of DNA? Okay. Base pairs in DNA, adenine and thymine are going to actually have only 2 bonds between them. Okay, cytosine and guanine are going to have 3 bonds between them. That is true. Okay, what about this one over here? Again, with the same one. But the difference is the, orientation. This orientation is wrong. It's supposed to be that one is up, 5' to 3' and the other one's on the opposite direction. So this is the correct answer. Okay, the diagram shows the process of transcription of a gene in the nucleus of an animal, which row correctly identifies P, Q, and R. P, Q, and R. Okay, so P and RNA polymerase is right here. So what is P is supposed to be, I think, the mRNA being produced, okay? And then Q is the DNA that's been opened. So we have, obviously, because we're transcribing this, the transcription is going to happen to the complement or to the copy or to the transcribed strand, because it's going to be, to the template strand, because the template strand is going to be the one to the template strand, because the template strand is going to be the one to be copied, because the mRNA should be basically a copy of the exact actual strand, the coding strand. So mRNA, it's not really mRNA, it's really the primary transcript, because it still hasn't undergone the changes that it needs to go through. So primary transcript, and Q is going to be the template strand. So and the R is going to be the non-transcribed strand. Okay. Which row correctly describes the role of growing leaves as sources or sinks for amino acids and sucrose? Okay, amino acids and sucrose. So this is the growing leaf. It's not going to be completely photosynthesizing and providing its own supply for itself and the rest of the plant. It's going to require some sucrose and amino acids from the roots and from the other organs or the other parts of the plant. As for amino acids, so same thing for both. I would say it's not completely producing its own supply. So let's see the options. Leaves act as sink, or leaves can act as sources or sinks. Okay, so there are multiple options here where the leaf has to act as one thing or the other. For both amino acids and sucrose, the leaf is relying partly on the rest of the plant, and it's partially also relying on itself. So it has to be both for both of these things. So leaves can act as sources or sinks. Leaves can act as sources or sinks, which is why it's D. Which row correctly matches each description to cilia or to root hairs? Contain vacuoles. Well, they're definitely not cilia. Okay, root hairs contain vacuoles. More than one represented per cell, definitely cilia. Okay, so root hairs and cilia. Which changes occur as amino acids are moved into the phloem, sieve tube elements at the source? Okay, the changes that occur as amino acids are moved into the phloem at a source. Yeah, so what happens to the water potential when amino acids are going to go to the source? The water potential should decrease. And so what happens to the volume of the phloem sieve tube? It's going to definitely increase, because when the amino acids go inside, they're going to decrease the water potential. So water from the xylem pits is going to come in, and it's going to start osmosing, or it's going to move down its water potential gradient into the phloem, increasing the volume inside. So it's going to be decrease, increase. Okay. The graph shows dissociation curves for hemoglobin and 2 different partial pressures of carbon dioxide, which the labeled point on the oxygen dissociation curves will result in highest concentration of hemoglobinic acid. Okay, so let's talk a bit about the pore effect. So remember, guys, first things, carbon dioxide enters the red blood cell. Okay, here it's going to react with water in the presence of carbonic anhydrase, and it's going to form carbonic acid. Now, carbonic acid is going to dissociate into hydrogen ions and bicarbonate ions. Bicarbonate ions are going to leave, and there will be the chloride shift, and hydrogen ions are going to stay, and they're going to react with oxyhemoglobin. They're going to kick out the oxygen, and they're going to bind to hemoglobin and form hemoglobinic acid. So when there is lots and lots of carbon dioxide, there's lots and lots of hemoglobinic acid. So you're gonna need high carbon dioxide partial pressure. Okay, so you're looking at the lower graph, and you're gonna need low partial pressure of oxygen, because this specific effect here, where like there's lots of carbon dioxide coming into the cell, where does this happen? It's gonna happen at muscle cells. It's gonna happen at respiring tissue. Respiring tissue do not have as much oxygen as lungs, for example, and that's why the partial pressure of oxygen should be low, not here, not high, because this is pretty much the partial pressure in the lungs. Okay, so it's B. Which statements about the transport of carbon dioxide into the blood are correct? Carbonic anhydrous catalyze the reaction of carbon dioxide and water to form carbonic acid. True. Carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions. Dissociates into hydrogen ions and hydrogen carbon. Yes, that is also true. Some carbon dioxide combines with hemoglobin to form carbaminohemoglobin. That is also true. Hemoglobin readily combines with hydrogen carbonate ions to form hemoglobinic acid. No. Okay, remember it's hydrogen ions and hydrogen carbonate. So it's 1, 2, and 3. What happens as a result of blood pressure in the left ventricle becoming higher than the blood pressure in the left atrium? Okay, so when blood is at higher pressure in the ventricle, to prevent backflow of blood to the atrium, then the left atrioventricular valve has to close. The left atrioventricular valve opens, the lunar valve in the aorta closes. Okay, Cellular valve in the aorta opens. Why it wouldn't be one of these two options? Because the first thing that happens is this one, okay? It has to be that the left atrioventricular valve closes. And then when the pressure in the ventricle becomes higher than that in the aorta, then the cellular valve is going to open to allow flow of blood into it. And then when pressure in the aorta is higher than that of the ventricle, then the cellular valve closes to prevent the backflow of blood, okay? So that's why it's A. Electron micrograph shows the inner surface of the human trachea. What is the identity of part of the electron micrograph labeled T? Okay, so this is the trachea. So you can see here that there are some ciliated cells and some other cells, right? So there's ciliated cells. So the other cells here are the goblet cells. So they're labeling T. Okay. So they could definitely be goblet cells releasing mucus. Okay, squamous epithelial cells. Well, they don't look like squamous epithelial cells, and if we're being honest here, we are looking at the human trachea. And the epithelium in the trachea is composed of pseudostratified columnar epithelium that is going to have ciliated cells and goblet cells, right? And so the ciliated cells are available, as you can see. So the goblet cell is the second option. Okay, which row correctly summarizes the typical distribution of cartilage and smooth muscle within different parts of the gas exchange system of humans? Okay. So cartilage and bronchi, bronchioles, trachea. So cartilage is found in bronchi and in trachea, but definitely not in bronchioles. Smooth muscle is found in... Okay, so what exactly? Oh, oh, they are telling you. Okay, so they're asking which rows correctly summarize the typical distribution of cartilage within... Okay, so correctly... Bronchi having cartilage and smooth muscle is true. Bronchioles having no cartilage and no smooth muscle, that's not true, because bronchioles have smooth muscle. Trachea having both is actually, well, true for the most. Yes, so it's 1 and 3. These are true. They want the true? Yes, the correctly. Okay, so it's 1 and 3. A molecule of oxygen diffuses from the air in an alveolus to hemoglobin in a red blood cell. Assume that the molecule crosses cellular layers by passing through cells rather than between cells. What's the minimum number of phospholipid layers that a molecule of oxygen can pass through? Okay, let's start. So first, in the alveolus, you're going to have... Let's draw. Okay, I always like to draw in this kind of question. So we first draw an alveolus. An alveolus has squamous epithelium. So you're going to have squamous epithelium here and then some oxygen. These are the nuclei, by the way, so you guys don't get confused. Anyway, oxygen is in the alveolus, and then it has to cross the wall of the alveolus in order to enter the blood capillary where there are blood cells. In the blood capillary, you have endothelial cells lining it. Okay, so you just draw endothelial cells, and then you have the red blood cell in here. Okay, and then you can close the blood capillary right there. Okay, doesn't matter. Now, the red blood cell, so in order for oxygen to go from here to there to here, how many phospholipid layers are we going to cross? And remember here, he asked you for phospholipid layers, not for the number of membranes. So first of all, in a membrane, there's a phospholipid bilayer. Okay, so here... it crosses one phospholipid layer, two obviously phospholipid layers. Let's just do membranes and then we're going to multiply everything by 2 because in each membrane there's 2 phospholipid layers, okay? So here we cross a membrane, here we cross the second, third, fourth, and fifth. Remember he said that we have to cross all cells. We can't go in the gaps between cells. So there's 5 membranes, so you multiply that by 2 to get the number of phospholipid bilayers, and that's 10. Okay. Which row shows the type of pathogen that causes cholera and its mode of transmission? Cholera is caused by Vibrio cholera, a bacterium. Mode of transmission, contaminated food or water. Definitely, cholera is a foodborne infection. Which statement about tuberculosis is not correct? Tuberculosis can be controlled by vaccination. That is true. There is a vaccine for TB. Tuberculosis is caused by a virus spread. Okay, that's not correct. It's caused by a bacterium called Mycobacterium tuberculosis or Mycobacterium bovis. So B is the wrong answer. HIV or AIDS increases the risk of developing TB. That is true because tuberculosis is an opportunistic infection, meaning it waits until the immune system is weakened so that it can actually do its thing and attack. And so HIV and AIDS, they are basically they're like things that weaken the immune system, and so definitely TB may be transmitted by eating contaminated meat. 100% Mycobacterium bovis is found in contaminated meat. 37 more cases of malaria are being reported in Europe. Other diseases formerly confined to tropical countries and transmitted in the same way as malaria have also spread to parts of Europe. Tropical countries have higher mean temperatures and humidity than Europe. What could explain the increasing number of cases of these diseases in Europe? Okay. Malaria is generally found in places near the equator, and so Europe is generally not one of the places where you would find malaria. If there's increase, it could be maybe increased travel between these countries and Europe, which means that we can have insects in the airplane and people with malaria coming into these countries and spreading the infection. Obviously, malaria is spread by vectors, so there should be increase in mosquitoes in the country itself as well, and that could be because of maybe increased climate change or something of that sort here. Rising temperature and humidity in Europe as well as a result of climate change, 100%. If there is increased climate, that's a better environment for our vector, which is the female Anopheles mosquito. Increased travel between Europe and tropical countries, 100%. Creation of wetland areas such as marshes and a to increase biodiversity, 100%. Wetland areas because mosquitoes lay their eggs on wet areas, and so if there's more wetland areas, like in the tropical regions, then there's definitely more eggs being laid and more female Anopheles mosquitoes and more vectors, and so more spread of infection, and that's A. Moving on to 38, the diagram shows 3 different bonds. Here you can see a hydrogen bond in number 1, number 2 you're going to see a peptide bond, and 3 you can see a disulfide bond. Which bonds are found in an antibody molecule? Okay. An antibody molecule has disulfide bridges actually, so 3 is available. Okay, 2 is also definitely available because it's a protein. And then hydrogen bonds are part of the secondary structure, so you're probably going to see it too. And that's why I'm going to pick 1, 2, and 3. Okay, specific monoclonal antibodies can be used to treat some of the forms of cancer. One example is trastuzumab, which can be used in the treatment of tumors caused by cancer. Okay, which statement could explain why monoclonal antibodies are suitable for this role? The variable regions of monoclonal antibodies such as trastuzumab can change, allowing the antibodies to bind to cancer even if the antigens mutate. That's not true. Okay, the monoclonal antibodies variable region is going to stay yes, it's variable because it is going to be different for each different kind of antibody, not because it can change. Monoclonal antibodies such as this one bind to specific cell receptors on cancer cells, which prevent these receptors from receiving signals that are needed for cancer cells to grow and divide. Or the antigen binding sites of monoclonal antibodies such as trastuzumab are complementary to antigens found on some cancer cells and have stable tertiary structures that do not change. Exactly. Okay, so they have similar antigens to the ones on cancer cells, so they can bind them, and they can lead to inflammation, and they can lead to leukocytes and white blood cells getting rid of them. Okay, and then 2, monoclonal antibodies are going to have specific cell surface receptors on cancer cells. I mean, that could be a function, that could be a role, so why not? Okay, but obviously the only one here that does not make any sense is going to be 1, which is why your answer is 2 and 3. HIV virus causes an illness by infecting and destroying T helper cells. This leads to AIDS and an inability to produce an effective immune response. Which component of immune system are produced less effectively when AIDS develops? Okay, when AIDS develops, as they've mentioned here, it destroys T helper cells, and so this leads to AIDS. Causing, as I mentioned, destroying an effective immune response. So the main thing that's going to happen is going to be that memory cells are going to be destroyed pretty much, and then obviously when plasma cells are not going to be produced as much because there's no B memory cells and no B cells, and so when there's no B memory cells and no B cells, there's no plasma cells because plasma cells are derived from them, and antibodies are produced from plasma cells. So it's really 1, 2, and 3 is your only option. And that's it for this test. I hope this video was useful for you guys. If it was, make sure to share this video with anyone who might use it, and I'll see you guys in my next one.